started. I'm definitely not gonna go full 2 hours today, especially in this environment. But we'll get our feet wet a little bit with the bacteriology block that we're starting today. And we'll start off with probably, or at least arguably, one of the most significant bacteria that you will encounter. It's very frequently encountered. You've probably all heard of Staph aureus. So it deserves its own deck. We've already, it was a question on the exam, it wasn't about the Staph aureus, but you've at least seen it a couple of times. And increasing it and trying to expose you to some of these common bacterial names, just to get you used to them. But here we are, block 3 is upon us. Here's what we're doing for block 3, 4, and 5, basically. Just basically looking at all these different pathogens and talking about what their morphology is, how they're transmitted, how they cause disease, pathogenesis, their virulence factors, some of the epi, how they're spread to your patients potentially, and how we work them up in the lab. A little bit finer detail for block 3, talking about, so these will be the same for all the decks in block 3, which are organized, by the way, according to the gram stain and the morphology. So the first deck is GPC's staph, gram-positive cocci staph. Second deck is strep, because it's that important, also is gram-positive cocci. And then by the third deck, we're finally ready to move on to some gram-negatives, but mostly talking about the enteric bacteria. So we're going to be spending a couple days talking about diarrhea. It's going to be awesome. And then we'll finish off with some more gram-positives, some gram-negatives. But that's kind of the way traditionally bacteriology is arranged by that gram stain, because that's directs, at least initially, how you choose antibiotics. It is important. It's not some random thing. It is important. But going back to the ELOs here briefly, we will, depending on the organism that we're talking about, we will go into more detail on the more important ones, which I'll kind of lead off with a little bit in more detail the first couple slides. But if it's something like Staph aureus, we're going to go into a lot of detail because it warrants the attention. If it's something less important, we will spend less time talking about things like its morphology, etc. One of the biggest objectives of this entire course, as we're going through all these pathogens by name and talking about them, is to get you familiar with the names of the bacteria, or viruses, or parasites, or fungi, and the types of diseases they could cause in your patients. So that's one of the big things we want to do. And then to a lesser extent, we'll talk about appropriate specimens and how we work this up in the lab as needed. Right, so here we are. Get my laser pointer out. This sort of just orients you. I started using this flow chart as sort of an intro slide for a lot of the bacteriology deck, just to keep you abreast of where we are. I did color code it, but that's okay. You don't need to be able to see this color scheme. So we're talking about bacteria, and not going to talk about gram-negatives until we get to the third deck. So we're talking about gram-positives, not talking about rods, just cocci for the rest of this week probably. And then when we're talking about gram-positive cocci, or GPCs, there are 3 pretty important genera, 2 super important genera, Staphylococcus and Streptococcus. So today is just Staph, and about, well, the vast majority is Staph aureus, by far the most clinically important member of this genus, Staphylococcus. But also significant is Epidermidis and Saprophyticus, and some others as well, but these are the 2 primary other clinically important staph species. In terms of why, as I alluded to earlier, why we spend so much time talking about things like E. coli and staph and strep, it's because when you enumerate all the different types of bacteria that are isolated in clinical labs across the country, let's say like an annual basis, there's a lot. There are hundreds. There were about 534 different types of bacterial taxa that were reported. So a huge variety of bacteria can cause infections in patients. But there's a lot of repeat offenders, so those are the ones we want to spend our time studying and getting to know really well. If you look at this graph here, it shows what percentage of all these 534 isolates are broken down according to how commonly they're discovered. I didn't express that very well, but For example, if you follow up, 90% of these isolates belong to just a relatively few number of taxa, 16 different taxa. Remember when we were talking in block 1 about taxa? What a taxon is? What's a taxa? It's any echelon of taxonomy. So yeah, but close. So this could be a genus, it could be a species, but 16 different taxa. And then if you just bump that up a little bit, include another 9 different categories of bacteria, you get all up to 95% of isolates. So we don't need to study that many in order to get a good sense of what you guys are going to be seeing most often when you start seeing patients. But that said, one other point I want to make is we still need to study some really rare stuff too. And that might drive you crazy, but it's important. Like Bacillus anthracis, for example, where do you think that's going to be on this chart? How often do you think you're going to encounter anthrax? It's going to be like way over there. You're never going to see it, right? Francisella tularensis, not see. Yersinia pestis, right? But we still have to talk about them because you can't go into practice, especially being a military provider, and not know about those biowarfare agents as well. To kind of drive home that point in terms of the frequency of recovering these different bacteria, E. coli is probably number 1, frequently recovered. But second place is Staphylococcus aureus, an Enterococcus species, which we'll be talking about in the next step. Coag-negative staph species like Epidermidis and Saprophyticus, so on and so forth. We should be covering all of these guys. These are common, very commonly isolated and recovered bacteria. Okay, so gram-positive cocci. We know that gram-positives, just as a general feature, have in their cell wall a very thick layer of peptidoglycan, right? What kinds of traits does that confer to bacteria like Staph aureus? Well, if you have a thick layer of peptidoglycan, that makes you relatively resistant to drying. These guys are also pretty hardy in other respects as well. They can tolerate pretty high salt concentrations. And you have a pretty high salt concentration on your skin, so they can survive on your skin. They are also facultative anaerobes. What does that mean? Facultative anaerobes?
Someone.
Yeah, can survive in the presence or absence of oxygen. Because of that, they can survive either on your skin or deep tissue infections, anaerobic environments like that. The plate on the bottom just shows that they are expressing some hemolysins, which you guys definitely did well in this morning. That question, you know what hemolysins are. Those are types of lytic molecules that can break down membranes. So you can see that zone of clearing here. That's very typical for Staph aureus, especially for virulent strains of that Staph aureus. Up at the top, we can see a gram stain. Very clearly see gram-positive cocci. They're kind of on the big side. They're not hard to see. 0.5 to 1.5 micrometers in diameter. Usually pretty spherical, but they can be a little bit, a little squished sometimes. The fact that they grow in clusters is also pretty distinctive. When you're a microscopist or lab tech and you're reading a lot of gram stains, and you see gram-positive cocci in clusters, it's a good chance it's going to be Staph aureus. But we know gram staining isn't definitive, but it's indicative, and that's because of the way they divide. Unlike bacteria that divide and grow in chains, and we divide in one plane essentially, these guys, where they divide, they'll divide into different planes. Because of that, you get these tetrads and clusters that form. So when we get a gram stain, this kind of just walks you through some of that initial thought process that is valuable to the lab when you're sorting out what's what. You get gram-positive cocci. If they're in chains, they're probably going to be something like Streptococcus, maybe Enterococcus, but likely not Staph. However, you could do a test called a catalase test that would help determine whether you're dealing with strep or staph a little bit more definitively. Most staph species, because they are facultative anaerobes, they need to be able to produce this enzyme called catalase. Anybody know what catalase does? Made a little mistake on there. It should be, the verb is catalyzes, but catalase catalyzes. What does catalase do? Outstanding. That's really good. Okay. Yeah, so there's a test for that, but this allows that particular bacteria to be more proficient at breaking down those free radicals in oxygen, so that it survives oxygenated environments better, and that's a big advantage for these bacteria. So the test for catalase production is you put a little bit of that bacteria, once you've isolated it on a colony, you drop a little bit of hydrogen peroxide directly on that colony, and if it bubbles, as in it releases oxygen, it's a catalase producer. It's catalase positive. That would be an indication that you're dealing with staph and not strep. From there, the next question is it Staph aureus, which is the most clinically significant one, or is it something else, one of those coag-neg staphs? So, coag-neg staph, it's kind of another spoiler alert. We're looking for production of coagulase, which you guys know well, does what? As a virulence factor for those bacteria, it
Makes a ball around it.
no, that would be what?
Agglutination.
Granulation, that's right. No, so
Found
coagulase.
that one out.
Yeah, you did. So what coagulase does is it helps form basically a clot or barrier around that bacteria, and it prevents it from being phagocytized. Things like macrophages and neutrophils. So when those coag-positive Staph aureus species are producing coagulase, you can do a couple different tests for coagulase production. You can do a tube test or you can do a slide test, but both have the same principle. If they produce coagulase, the coagulase will convert soluble fibrinogen to fibrin, and you get like a clumping reaction going on. If it's a coagulase negative species of staph, you don't get that coagulation reaction, and it remains liquidy. So this is the test tube coag test, but you can also just put that like a bacterial suspension of staph on a slide, and then you could drop some rabid plasma on there, and if it is Staph aureus, then it will clump, it will coagulate together. And if it's like Staph epidermidis, it will not. Okay. In terms of where this bacteria likes to live, like I said, it is hardy, it does well in a lot of different environments, so it can colonize all over the skin, but its favorite place to be, number one favorite place is where? In the nares or in the nostrils, the anterior nostrils, that's where it likes to be. Even then, when folks are colonized with Staph aureus, it's a fairly significant portion of the population, but some of these people are colonized for long-term with Staph aureus, some of them transiently, and some people are just non-carriers. They don't really have staph in their nose that often. So it depends, and that's fine. We talked about bacteria being in a certain niche and not being a problem, not being pathogenic. So generally speaking, if you have Staph aureus in your nose, even if it's MRSA, whatever, that's fine as long as it stays in its niche. But we'll talk about times where it can get out of that niche and cause problems. Because of that hardiness we talked about, its resistance to drying, that big, thick peptidoglycan layer, it does well on fomites, so if you are colonized and you are not washing your hands and your finger goes up your nose perhaps and then onto something, that Staph aureus, when it is transferred, it'll stay there in the environment for a while and it can be transmitted. So fomite transmission of Staph aureus is definitely a thing, and especially in hospitals, this is where you need to be cognizant of the fact that it can survive on inanimate objects for a while. Contamination of food. Most people don't really associate Staph aureus with foodborne illnesses, but this particular bacteria, it does produce exotoxins, and those exotoxins can cause problems with foodborne illness. So we'll talk about that in a little bit more detail. That's another way that you can spread not just to another person, but to many other people in big outbreaks. Anytime you get foreign bodies introduced into your body, that is a major risk factor for introducing Staph aureus where it should not be. Anything from a mild little splinter to a shunt or a catheter or shrapnel, whatever, increases your risk for acquiring Staph aureus. Talking about some of these virulence factors, at this point you've heard about these before, but so you should be pretty familiar familiar with most of these virulence factors, but not all bacteria have them. These are specifically some that Staph aureus have and that they use in order to cause infections, serious disease. Capsule we all know about. With Staph aureus, the capsule that's produced is a very thin capsule layer of polysaccharides. That makes it, of course, difficult for them to be phagocytized. Most of these clinically significant isolates are going to express capsule protection. They're going to have capsule. So that tells you that it's important for them to survive in the host. Protein A, we've talked about that before too, right? That should hopefully ring some bells. What does protein A production do for the bacteria? What does it bind? The Fc region of antibodies. So this figure shows that really well. Here the protein A would be that orange V-shaped molecule on the top of this toxide. When you have it coating the exterior of the bacteria, it will bind to the Fc region and prevent these macrophages or neutrophils from engulfing or phagocytizing the cell. And then we talked about adhesive surface proteins before, your miscrems that will bind to things like fibrin, collagen, laminin, extracellular proteins, able to attach and establish colonization of different surfaces in the body. Getting into toxin production, we talked about exotoxins and general qualities of exotoxins. Here is an example of a bacteria with Staph aureus that can produce a lot of different toxins. Not all strains produce all of these toxins, and that has an effect on what kind of disease it's capable of causing. But these are just some of the toxins that are associated with Staph aureus. First and foremost, cytolytic exotoxins. We have 4 different types: alpha, beta, delta, and gamma. As the name suggests, what these toxins do is they bind to cell membranes and they cause target cells to rupture or to lyse. Alpha toxin is probably the most significant of these 4 different cytolytic exotoxins, but they're all important, and they all also have hemolytic activity. So when we're talking about when you culture out Staph aureus on a plate and you see zones of hemolysis around those colonies, some of those factors that are responsible for those zones of hemolysis are these cytolytic exotoxins that can also rupture erythrocyte membranes. Another type of exotoxin that works similarly to alpha toxin is this PV leukocidin, Canton Valentine leukocidin. This carriage of this particular toxin is associated with some of the more aggressive and virulent strains of Staph aureus. So the strains that are isolated for patients who have really terrible necrotizing pneumonia infections, or awful skin and soft tissue infections, oftentimes they will produce this very damaging leukocidin, which has a more restrictive range than those cytolytic exotoxins. This one specifically targets things like macrophages and neutrophils, and it destroys them. We also have some toxins that act as superantigens. This slide is also a review. We've talked about this before. But with superantigens, we're talking about an inappropriate and constant stimulation, particularly of CD4 cells. When our antigen-presenting cells, this is a busy slide, but that's okay because it's representing like the chaos of superantigens. So we have our antigen-presenting cells, and they're interacting with this T cell in the center there in yellow, and that T cell is just constantly being stimulated. So it's producing all the cytokines that we talked about before, IL-1, IL-2, TNF-alpha, interferon gamma, and it's causing just massive overstimulation of the immune system as a result. That is because these exotoxins that have superantigenic properties can bind outside of that normal groove in between MHC class II and your T cell. So, but here on the right, you're seeing that superantigen is binding outside that groove in a non-specific way. That's what's causing that effect. Some examples of Staph aureus exotoxins that have superantigen activity are these enterotoxins. There's a bunch of different enterotoxins. These 6 are all important, particularly these first 4. They're called enterotoxins because they're associated with food poisoning, as the name suggests, but some of them are not necessarily restricted just to food poisoning cases. We'll talk about this more in a couple slides, but the thing that makes these enterotoxins so dangerous, particularly when they are found in food, is that relative to other exotoxins, they're fairly heat stable. That was one of the defining characteristics of exotoxins, is that they are not as heat stable as endotoxins. But in terms of exotoxins, these guys can withstand a little bit of mild heating. Not thorough cooking, but like nuking something in the microwave to reheat it for 15 seconds probably won't deactivate these enterotoxins. So they retain that superantigen activity. And they have that non-specific excitation or stimulation of the immune system on top of another specific activity, and that is stimulating this vomiting center, the triggering this vomiting center in the brain. So that has the emetic effect when you have like a really sudden onset foodborne illness. It just causes you to violently start vomiting. Another toxin produced by Staph aureus that has superantigen activity is toxic shock syndrome toxin 1. There is a 2, but we're only going to talk about 1. TSST1. We'll talk about this more as well. It was originally called enterotoxin F for historical reasons, but as it became characterized and associated and linked to toxic shock syndrome, like menstrual associated toxic shock syndrome, they renamed it to TSST1. Okay, but structurally, it's very similar to these enterotoxins. And finally, there's exfoliating toxin. This has also some specific activity combined to this target called desmoglein, which is a factor that's associated in the epidermal layers, and it helps keep the integrity of the skin together. So when this is targeted, you get a really nasty peeling of the skin and sloughing off of those outer skin layers. We also have, in addition to those toxins, some invasive enzymes that we've talked about before that can contribute to some tissue damage. Yes, sir.
Ma'am, when you were to see something like SRS strain, is it just going to say SRS strain A? SRS, like, how is it like written out, like for us to like visually see it? Like if it was a test question or something like that, how would it be annotated or written to say?
Oh, we will not get into like specific strains for Staph aureus. Yeah, just know that it's like some strains carry it, but we're not talking about any particular strains of Staph aureus. Okay, so Staph is kind of well known for its ability to cause skin and soft tissue infection. And in order to be able to do that, it has to possess a lot of these invasive enzymes that we've talked about to varying extents. These skin and soft tissue infections, we'll spend a couple slides talking about it just because we have to, just in order to understand what they are, but you will cover that more in other courses. Basically, we just want to illustrate that these skin and soft tissue infections, they can range all the way from really mild stuff like folliculitis, inflammation of the hair follicles, all the way up to deep tissue infections. So there's a wide range. A lot of what determines that range is how the bacteria are entering into the sites, like if there's a puncture wound, for example, versus just your normal skin in the Staph aureus that happens to be making its way down into the hair follicle and causing like inflammation around that area. Going back to these invasive enzymes, so we've talked about lipase before. Lipase is responsible for breaking apart some of those phospholipid barriers, so it can, like around that hair follicle, for example, this is a pretty well-protected structure. But with lipases being released locally into that area, it will degrade and loosen up that area and allow those bacteria to kind of come in and settle in a little bit more deeply, allow them to colonize a little bit more. Hyaluronidase, as the name suggests, it breaks down hyaluronic acid, which is a very, very common connective tissue in the body. So that too plays a major role in establishing invasive infection. Coagulase, just to the extent that it protects those bacteria from like local tissue macrophages, resident macrophages, if need be. And then when the time is right, fibrinolysin, as we know, it has like the opposite effect of coagulase. Coagulase will allow a clot to form around the bacteria. When you start producing fibrinolysin, or the specific form of fibrinolysin called staphylokinase, that allows bacteria to spread on their own terms when they are ready to do so. So it gives them control over when and how they disseminate and spread. And then finally, nucleases like DNases. This will help them contribute to biofilm formation, which if you remember back when we were talking about disinfection and sterilization in Block 2, these guys are serious biofilm formers. So the ability to form these nucleases will help them, it will contribute to their cycles of biofilm formation and release. We'll talk about that too a little bit more. Okay, so those are some of the invasive enzymes that are produced. With biofilm formation, I kind of blew up this cartoon a little bit to show how this works, but let's skip down to the significance and we'll talk about those steps. Why might some types of bacteria like Staph aureus and like Pseudomonas aeruginosa, why might they need to form biofilms? Yeah, so it's kind of like a shift in mentality from the individual to the community. In theory, the guys at the bottom of this massive pile are going to be better protected from things like antibodies. And what else? Like your own immune system, and what thinks about some artificial things. What else? Disinfectants. And then what about if you just want to treat that patient with penicillin or some other antibiotic? And that antibiotic, it might be able to possibly get through some of that matrix and sticky junk on the exterior, but is it going to be able to necessarily penetrate all the way down to the bacteria they're seeding when it's layers and layers and layers deep? So very protective. We've got host defenses, we've got protection from antibiotics. Very important in the ability to allow for chronic and recurrent infections, and that goes to the cycle that can sometimes happen. With Staph aureus, we know that you have an initial attachment to some sort of substrate, whether it's bone or plastic or metal or whatever. And then you have a seeding and proliferation where the cells start dividing and they can grow up. Once they reach a critical concentration, we have a form of cell-to-cell communication called quorum sensing, where they start producing small molecules, or basically hormones. And those hormones are signals that are received by other bacteria that tell them to turn on genes that allow them to have biofilm qualities. So again, this is a shift to more of a community mentality, and it activates a lot of different genes. A lot of different genes about survival, nutrient processing, metabolism, all sorts of things, and expression of all of these extracellular substrates as well. So this biofilm will start growing up. Sometimes it's just straight staph, sometimes it's polymicrobial and has a bunch of different types of bacteria and it's very complex. And it will reach sort of like a critical mass where it can stay that way for a while, but if it no longer needs the protection that's conferred by that biofilm, if conditions become favorable, it can start activating genes that permit like the dispersal of some of these bacteria on the outside. So these individual bacteria that will start breaking off from the biofilm to go and reseed in a new area, those are called planktonic bacteria or free-living bacteria. But again, this is when things are good and it's favorable to go explore and venture and try and find some more hospitable area, someplace where there are nutrients and things like that. Yeah.
And when you were talking about activating specific genes for a variety of things, was that happening in proliferation or maturation?
Probably during both, yeah. But there is, like, quorum sensing is directly related to reaching a critical concentration of that signal, which is a hormone that is secreted from bacteria to bacteria. Once they have a critical concentration, all those inducible genes will start turning on to tell them, like these pro-biofilm formation genes will start turning on. Yeah. Okay, so what types of diseases can be caused by Staph aureus? We've already mentioned a little bit about the skin and soft tissue infections. That's what SSTIs stand for, skin and soft tissue infections. Very common acronym. But some others you might not associate as much with Staph aureus. We've got pneumonia. We've got infective endocarditis. We've got sepsis. Bone infections, osteomyelitis. We'll talk about all these in a little bit more detail. So if you've never heard of these before, that's fine. We'll go over them. The commonality with all 5 of these different categories of infections that I just mentioned is that they are true infections. Those are caused by the bacteria themselves, growing, colonizing, surviving. Contrast that with this group at the bottom of your screen, toxin-mediated diseases. You've got toxic shock syndrome, you've got scalded skin syndrome, and then you've got food poisoning. These diseases can affect you regardless of the bacteria are there anymore or not. Probably the best example is food poisoning, where you have, like, I don't know, your pizza from last night, you nuked it for 5 seconds this morning. You might have killed the bacteria, let's say it had Staph aureus on it, but you did not destroy the enterotoxins that were on your pizza. So if it makes you ill, if you consume enough of the enterotoxin to make you ill, that's a toxin-mediated disease, no bacteria are present, and you're not going to find or isolate any bacteria from that patient, it's all toxin-mediated. These other examples are similar. There might be like a localized foci of bacteria, but the effects are usually systemic because this bacteria has started producing exotoxins, and exotoxins are small little proteins that can be swept away by circulation and they can have far-reaching systemic effects. Ooh, a lot of text on this slide. Localized skin infections. Couple terms here. One is pyodermas, that's a good one to know. Pyodermas are simply those pus-like skin infections. Suppurative skin infections, what does the word suppurative mean, do you think? Maybe. You might have said it. Say it again just to read. Oh, what? That's a good guess. It's actually fairly synonymous with the word purulent.
Pustules?
Yes, yes. I was going to say that begs the question, what does purulent mean? But yes, pus-filled. So suppurative skin infections are pus-generating infections. There's pus involved with this. Pus, as we know, is just a collection of neutrophils, dead bacteria, dead tissue. So we've got pyodermas, other suppurative skin infections. We've talked about folliculitis a little bit before. Sweat and sebaceous glands, the glands themselves can become invaded and inflamed. What's a sty?
Like affect the hair.
Yeah.
follicle, right?
Yeah, usually just the eyelash itself will have some bacteria. Very common for those styes to be caused by Staph aureus, but other bacteria can cause styes as well. So it inflames the eyelash. Furuncles, anybody ever seen a furuncle before? Yeah. What does that stand for? Could be. I haven't heard that term before. But yeah, it might be. A furuncle is one of those suppurative infections. So it is pus generating, and it affects a little bit deeper than these other guys. So we're going now deeper into subcutaneous tissue. Should have put it in this slide deck, but for the STEP deck, there's a nice picture that shows the layers of the epidermis. Have you guys had that in AP yet? You talked about the skin. Okay, so you're going to be masters of where this is happening. Cool. Okay, so that's how deep furuncles go. They go into those subcutaneous tissues, and abscesses are simply pockets of pus and localized bacterial infections. They can be really small, like a pimple is technically an abscess, or they can be massive and devastating. So, but an abscess is simply just that, a localized bacterial infection. Carbuncles. What are carbuncles? Never heard of them? These are like when furuncles start coalescing together and they get bigger and deeper and worse. So carbuncles are like really bad furuncles. When you start getting into the carbuncle range, you will probably need to get some treatment for that. We might be talking about, in addition to draining the pus out of that area, perhaps antibiotic administration, topical, maybe even oral antibiotics in order to clear that up. So on this list, carbuncles would be the most serious one. Impetigo, if you've ever heard, anybody heard of impetigo before? A couple people, okay. Impetigo is not as serious as carbuncles. What kind of folks usually get impetigo?
Kids.
Kids, yeah, exactly. So impetigo, just go to this next slide. this is what it looks like. It's kind of that honey-colored crust that forms usually around the mouth or the nose, mucosal area. And it is associated with poor hygiene practices, so like wiping, snot all over your face, things like that will eventually, if it has staph, some of that staph can get into that skin, and you get that crust. But it's usually not terribly severe. Here's a picture of a sty, type of folliculitis we talked about. That looks like it's very painful. And it's usually treated with just like a warm compress, unless it's really bad. Folliculitis it can occur on any hair follicle, but usually you get those on the face or on the neck, scalp, groin, armpits. Here's your furuncle, where you can see it's separating. It actually has that yellow, pussy center, that boil that's coming out. And then cellulitis, we haven't talked about. We'll talk about cellulitis a little bit more when we talk about strep pyogenes. But anybody know what cellulitis is? What do you think?
It's a more superficial skin condition, generally not like an operation or an operation site, but it can be really painful, spread really quickly, it's very erythematous.
Yeah.
Touch.
Yeah, so this is going, it doesn't look that bad. Here you can see there's a focal point, meaning you can see like that there's a break in the skin, but the infected or affected area is much larger than that focal point. If we were to look a little bit deeper into that tissue, we would find destruction of some of those deeper layers. So this is going into that subcutaneous tissue, not quite to the extent of the fascia, but getting pretty deep. And just like he said, it is erythematous, meaning what? Red, yeah, and it's hot to the touch. So you definitely have a pretty nasty infection. Those are serious infections. They may not look like much, but they are serious. And they'll get much, much more serious than that. Abscesses, like I said before, this is a really bad one, but they can be minor. You see how deep that is, and it looks like that blackened tissue on this person's hand is suggestive of what? Necrosis, yeah. So that tissue looks like it's starting to die. We have some deeper localized infections, so we're still not talking about systemic infections yet, but when we're talking about septic arthritis and osteomyelitis, this is when these bacteria really penetrated deep into sites that should otherwise not be accessible. So bacteria that can infect and invade bone, they usually get there by circulation. That's what hematology means, that they can be picked up by circulation and spread into those sites. By virtue of those adhesins, again, they're able to attach to bone and start colonizing and expressing some of those invasive enzymes that will ultimately potentially destroy some of that bone. With arthritis, too, some cases it starts off with a case of like cellulitis nearby a joint, and those bacteria will make their way over to the nearest joint if it's like an elbow, and they'll start replicating and dividing, and you get a lot of localized inflammation. You can get destruction. Hyaluronidases will start destroying cartilage in those joints, and so you get some pretty bad arthritis. And for that, to diagnose, you can do some imaging studies. You can see some of that cartilage damage on MRIs and X-rays sometimes. But if you want to send it off to the lab for ID, you need to aspirate that synovial fluid of the joint and send it down to the lab. Acute endocarditis. This is an infection of the inner lining of the heart, endocardium. Again, not something you would normally see with healthy people, but in all of these causes of acute endocarditis, they're associated with somehow a contamination event. With these examples all involving needles. The first is when a needle punctures your skin and it's delivering a contaminated product, like a medicine that's been contaminated with Staph aureus. The second is when you haven't properly disinfected the skin. You're supposed to put an antiseptic on the skin. So you get just normal colonizing skin bacteria that make their way directly to the bloodstream and ultimately end up in the heart. And then the last one are needles themselves, that, like the needles are contaminated by the bacteria. This happens usually when you're sharing needles, so it's more associated with drug users than it is in hospital settings. With pneumonia, You can have varying degrees of severity with Staph aureus pneumonia. This really serious form where your lungs are just kind of rotting away, severe necrotizing pneumonia. The isolates that they've been pulled out of those cases do tend to have expression of that one toxin I was talking about, that Panton Valentine leukocidin toxin. You can get pneumonia a bunch of different ways. It is possible person to person through droplets. If somebody is a carrier, particularly in a hospital environment, and they're breathing on a patient, they can transmit Staph aureus that way. It's also common as a secondary bacterial infection, especially after someone's recovered from a viral upper respiratory illness and their airways are already kind of softened up for infection. So in that case, it becomes more of an opportunistic infection. And then there's a form of pneumonia called aspirational pneumonia, which refers more to how you acquire it. What do you think aspirational pneumonia is?
Swallow a little food.
Yeah, could be food. It could be you actually don't really swallow it. You
Goes down here, wrong pipe.
you aspirate it and it gets directly into your lungs. So food, saliva, water has Staph aureus in it. It gets put directly in your lungs. Adults don't usually have this problem. Sometimes, it goes down the wrong pipe or whatever. But the people who generally suffer tend to be young kids, adults, people who have other issues with aspirational pneumonia. Ventilator-associated pneumonia, this generally happens in hospital settings, hospital-acquired or nosocomial infections. When people have been on ventilator support for around 2 days or more, 48 hours or more, they start becoming susceptible to VAP, ventilator-associated pneumonia, of which Staph aureus is a big cause. And I think this is, I think we'll end on this slide today. I'll let you guys go after this. So we'll talk about septicemia a little bit. This is an older term, but we need to kind of understand some other terms before we talk about septicemia. You've probably heard of sepsis. In fact, we've talked about it before in bacteremia. But what is the difference between somebody who has bacteremia versus sepsis?
Sepsis is the immune hyperresponse to bacteria.
Exactly.
Super antigen cytokines for all the intestines.
Perfect. Yes. So with bacteremia, we're talking about what exactly?
You got the bacteria in the bloodstream.
Exactly. So we don't describe how long it's been there. It could be very transient. In fact, when you get a splinter and that splinter has Staph aureus on it or some other bacteria, technically speaking, you have bacteremia, right? It's just a very fleeting thing. Your immune system will take care of it, no problem. It always does. But it's still a transient bacteria in your bloodstream. When it gets really bad, that can lend itself to sepsis-like conditions. The more bacteremia you have, the more bacteria in your bloodstream, generally that's going to trip off more and more of your immune system, and you can start getting into these sepsis conditions, which is just like you said, an uncontrolled dysregulation of the host response. So a lot of cytokine signaling, a lot of inappropriate inactivation. A few more terms, known focus, like you have a joint or a septic joint or a puncture wound or a surgical site infection in SSI. Or if you have no idea, we call those occult sources. Nosocomial infections aren't a type of infection, that's just a setting in which you acquire Staph aureus, but since Staph aureus is one of those nosocomial pathogens and opportunistic pathogens, you need to kind of be familiar with it and think of it also as a nosocomial infection. Very, very common cause of HAIs. Some of those are wound-related. They can be surgical site, like I said, SSIs, or they could be decubital. What is decubital? This gentleman has a decubital wound. Yeah, it's a pressure ulcer, yeah. So this looks like an older patient. This is a problem with a lot of older patients who aren't turned enough. They get pressure ulcers. And those decubital wounds that form can get infected and turn bad really fast. So these would be decubital infections. Catheter-associated infections, catheter-associated bloodstream infections have its own acronym. That's how common it is, CAPSIs. And in all of these, when you have these conditions that are bad enough where you get sepsis, they have a very poor prognosis. High mortality rates associated with those. Any questions about what we've covered so far?
Earlier, he asked a question, I can't remember exactly, but we talked about the enterotoxins, the labeling. So,
Strains? Is that the strain?
yeah, the strain question. So
Okay.
if you saw on a test, you'd be enterotoxin A or enterotoxin E.
Yeah,
Okay.
Yeah.
For labeling purposes.
yeah, and we'll talk about that early tomorrow too, like those enterotoxins, but yeah, some strains carry them, but we won't talk about. Yeah, those are the types of toxins, not the strains.
Right.
Yeah.
But it says strains right here, so it can be confusing.
Yeah, some strains of Staph aureus will carry different enterotoxins.
And the enterotoxins are subclass, enterotoxin A, enterotoxin B.
That is, the enterotoxins are just, they have different names and they have slightly different properties. So like enterotoxin B is really robust and it's associated with a certain type of strain is associated with certain types of food, etc. We'll talk about this stuff a little bit more tomorrow, but for all these different types of enterotoxins that are produced by different strains of Staph aureus, you really won't spend a lot of time asking you to differentiate them or know anything about them, with the exception probably of Staph enterotoxin B. That's the one that I want you guys to know. Yes, ma'am.
I'm just want to clarify for the exotoxin side, it only talked about alpha toxins, and we talked about some isocyanate and then alpha and do have plasmatic imbalance, other ones are
Yeah, the only reason why I was drawing, so that alpha toxin, that alpha cytolytic toxin is probably the most clinically significant one. And the reason why I was underscoring it is because it has that pore forming ability that is similar to PDL. The other cytolytic exotoxins like beta, gamma, delta, they can also cause hemolysis, but alpha toxin is the one that's most similar to PDL. Okay guys, you look super tired and I don't blame you at all, so I will let you guys go. If you want to come down to the office or go over your exam, you're welcome to do that, and I definitely encourage that if there's stuff that doesn't make sense from block. Okay, otherwise I'll see you tomorrow.